Hey guys, welcome to lecture number 1 of Unit 6, the molecular basis of inheritance. So at this point, you should have already watched a video. You should have done the mobile classics, the case study, and I'm just going to kind of quickly review a couple of things there, and then I'm going to jump into DNA structure and replication. So as you witnessed in your, or observed when you watched that video or went through the mobile classics, the main question in the first half of the 20th century was, in terms of science, genetic science, is what is the molecule of heredity? And as you remember, there were two potential candidates. There were proteins, which were deemed to be most likely the molecule of heredity because they're complex, much more complex than what they knew of DNA. They didn't really know much about the DNA. They didn't know the structure of the molecule, but they knew its components. They knew it was made up of 4 nucleotides, adenine, thymine, guanine, and cytosine. They knew it was in the unit of a chromosome, and kind of what they thought it was just a repeating unit of A, T, C, and G. And of course, now we know it's much more complex than that. And so they thought it was just too simple of a molecule to be the molecule of heredity. And the main betting money, was resting on proteins, but there were a few skeptics. Let me just close this out. All right. So again, that big question in the 1940s, what is the molecule of heredity? The two candidates, of course, were proteins and DNA. And as I said earlier, many people thought DNA was just too simple. Proteins, they knew, came in, literally hundreds of thousands of different combinations, and they knew that it was made up of amino acids. They knew there were about 20 amino acids that could make up a protein, and those proteins could each individual protein could be composed of thousands of amino acids. Well, they had watched chromosomes under microscopes in living cells dividing, and they knew that chromosomes played a role in heredity. But as you guys know, chromosomes consist of proteins and DNA. We have those protein histones that act as spools, which help to organize and pack the DNA into the cells. So there was protein in the chromosomes. And again, that's why the question was, is it protein or is it DNA? And sometimes they thought that maybe the DNA was acting as a way to organize the proteins to act as a molecule of heredity. So one of the very first experiments that was done in 1928, and they realized, of course, this was the Griffith experiment, and it was the discovery of transformation, of how genetic material, whether it was DNA or proteins at that time, not clear, could be passed from generation to generation. You went over this in your mobile classics, so I'm not really going to spend much time on it, but the main point being that the hereditary information can pass from dead cells to living cells. They call this Griffin called this the transforming factor. We are going to be doing a transformation lab in our classroom where we're going to take DNA from one organism and put it into another organism. That will happen in a few weeks. Okay, and again, that is transformation, the pickup of DNA from another source, generally outside of the cell, and bringing it into the cell and causing phenotypic change in that particular case. The Avery experiments conducted in 1944 were a way to determine what was Griffith's transforming factor, and was it protein or was it DNA? And they used a series of enzymes. They used DNase, which is an enzyme that basically breaks down the DNA. They used proteinases, which broke down proteins, and they used RNases, which broke down RNA. And when they basically added these various enzymes into different cultures, they found that the proteinases did not stop transformation, the RNA ACEs, which broke down RNA, did not stop the transformation process. However, the DNases, which broke down DNA, did destroy the transformation activity. So they concluded that DNA must be the molecule of heredity. However, this evidence was not enough for many of the hardcore believers that proteins were the molecule of heredity. So a little bit later, Avery, McLeod, and McCarthy came along, and they did another experiment. And in their particular experiment, once again, they basically concluded that DNA has to be the molecule of heredity. And this one was pretty kind of put the nail in the coffin, but still it was not good enough for many of scientists. So finally, what came along was the Hershey-Chase experiments in 1952. And this was the experiment that said that DNA was pretty much the molecule of heredity, and they were using viruses. Viruses are a great model or a great organism for testing this question because they are composed of the two candidate molecules, and that's it. They're composed of, or at least I should say, these particular viruses, which are phages, and phages are called, their long name is a bacteriophage. These are viruses that infect bacteria, okay, and they are made up of two things. They have a DNA molecule inside of a protein coat. This protein coat is called a capsid. And the way a virus works is it latches on, or where I should say the way a bacteriophage works, is it lands onto a bacteria and then it injects the genetic material into the bacterial cell. Meanwhile, the protein coat stays on the outside of the bacteria. It does not enter. So, Hershey and Chase utilized this information, and basically what they did is that they grew one series of viruses in a solution that contained radioactive isotopes of sulfur. And this is important because proteins contain sulfur, DNA does not, whereas DNA contains phosphorus and proteins do not contain phosphorus. So one group of viruses were raised or grown in a radioactive sulfur environment, whereas another group of viruses were grown in a radioactive environment of phosphorus. And so in this particular way, the ones that were grown in the sulfur had a protein coat that was radioactively labeled. The ones that were grown in the radioactive phosphorus had only DNA that was radioactively labeled. So what they did now was they then infected bacteria with one or the other of these viruses. Some bacteria were infected with the radioactive sulfur ones, and some were infected with the radioactive DNA ones. And then they basically allowed the viruses to replicate, the bacteria to replicate, and they looked at them. And what they saw was that the bacteria that were infected by the radioactive sulfur ones contained no radioactivity level at all, meaning that the protein molecule did not enter into the bacterial cells, whereas those that were infected by the radioactive phosphorus viruses, they contained radioactivity. So how did they do that? Well, basically what they did was they grew phages in, like I said, in a radioactive sulfur environment, the protein coats were labeled. They infected they infected the bacteria. They then put those into a blender, and all the blender did was to knock off the protein shells. And they're much smaller than the bacteriophages, or excuse me, they're much smaller than the bacteria. So they then put those into a centrifuge. The way a centrifuge works, of course, is it spins very quickly. The things that are heavier go down to the bottom. The things that are lighter float up to the top in what's known as the supernatant. And when they did that with this particular group, they saw that all of the radioactivity was in the supernatant and none was in the bacteria pellet, so to speak. Okay, this is all the bacteria that were infected. So the bacteria did not have any information inside of them. However, when they did this with the ones that were grown in radioactive phosphorus, the radioactive DNA was injected into the cells. They then put those into a blender, and then into a centrifuge, and the radioactivity was then detected down in the bacteria and not in the supernatant where the proteins were. And this was pretty much the nail in the coffin. DNA was the molecule of heredity. Now, look at the date, 1952. Watson and Crick did not come out with their model or publish their article on their model until 1953. So, I'm not exactly sure the timing of when this information came out and when it was available to Watson and Crick. I'd probably have to do a little bit of research on that. But anyways, if you want to watch a short video, it's like 3.5 minutes, just click on the link down here. I will post this presentation online. All right, so like I said, there was a lot of information already known and available to Watson and Crick. They knew that nucleotides were the building blocks of DNA and RNA, again described in 1869. The primary structure of DNA was determined in the 1920s. They knew that DNA was composed of these nucleotides. They knew it was composed, and each nucleotide was composed of a phosphate group attached to a 5-carbon sugar, which then had one of four nitrogenous bases: adenine, guanine, cytosine, or thymine, found in DNA. Or if they were looking at RNA, then they would have had adenine, guanine, cytosine, and uracil. Okay, so this is an important diagram to know. You look in your textbooks, you will see it also. Now, oops, let me go back. As you can see, when the structure was first determined, or I should say when Levine first determined the primary structure of DNA, he incorrectly stated that all 4 nucleotides were in equal proportions. And again, this led to the idea that DNA was simply not complex enough to be the molecule of heredity. And then World War II happened, and after World War II, a French chemist by the name of Erwin Chargaff found that the nucleotides actually occurred in certain ratios. Adenine was always approximately equal to thymine, and guanine was also approximately equal to cytosine. No matter what type of organism he looked at, he saw that same sequence of events. He saw that adenine was always equal to thymine in pigs and fish and humans and bacteria, and guanine was also always equal to cytosine in those same organisms. Okay, they weren't the same ratios in terms of, the same, fish and humans had the same ratio of adenine and thymine, or guanine and cytosine, but in each individual species, adenine always equaled thymine, guanine always equaled cytosine. Do remember, adenine and guanine are both purines, which are these double ring structures, and cytosine, uracil, and thymine are all pyrimidines. These are single ring structures. The way I like to tell my students to remember it is GOA, G-O-O-A, where the double O refers to the double ring structure. GUA is guanine and adenine. And TOC, T-O-C, which refers to with the the O being the single ring structure, and we have thymine and cytosine TOC. And now you could go UTOC if you want, or TOC-U, or something of that nature if you want to include uracil with that. But that's the way I have them. So GUA for adenine and guanine, and TOC or TOC-GU or UTOC for the pyrimidines. And remember, pyrimidines, big word, small structure; purine, small word, large structure. All right, so again, there is our nucleotide. And do remember, we talked about this earlier in the year, the way we number the carbons. This is, remember, the way we look at a structure such as this. Wherever there's a vertex, there's a carbon. We are leaving the carbons out, so it's easier to determine. It doesn't make it so confusing. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 of our 5-carbon or pentose sugar, whether it's ribose or deoxyribose. Okay, there's our 5-carbon sugar. Do notice that the first carbon is attached to the nitrogenous base. The third carbon is attached to a hydroxyl group, and the fifth carbon is attached to the fifth carbon is attached to the phosphate group. All right, so we had that information. Rosalind Franklin, of course, provided the X-ray pictures, which allowed Jim Watson to determine that it was a double helix. It allowed him to figure out how many different base pairs there were between each turn of the helix. He figured out there were 10. This is a base pair. Okay, we have a purine and a pyrimidine. That constitutes one base pair. Remember, we as humans have approximately 3 billion base pairs in our DNA, which is found in each one of our cells. Okay, whereas something like a bacteria may only have a few thousand base pairs. Okay, and a virus would have a DNA-based virus would even have even less than that. Okay. So Watson and Crick, there's their model that they built. We saw in an earlier picture, this is the physical model they did build. This is Jim Watson. This is Francis Crick. Jim Watson, I believe, is still alive. Francis Crick died a while ago. Jim Watson was much younger than Francis Crick. Jim Watson, unfortunately, has discredited his work in his later life due to his extremely racist views. To be quite blunt about it. Anyways, this was their structure. We saw that adenine and thymine formed 2 hydrogen bonds, whereas guanine and cytosine formed 3 hydrogen bonds between them. And the distance between the base pairs, and there was 10 base pairs between each full turn of the DNA. You can look at this diet. You can click on that if you want to see some more information about the structure. But once they had this model, the model explained Chargaff's rules. It explained Rosalind Franklin's picture. It explained basically all the information that was prior, and also gave information of how DNA replicates. Once they saw what the model was, they instantly saw how DNA could pass its information from one generation to the next. And then that's what we are going to be doing in lecture 2. So I'm going to stop this lecture right now, and we will come back and finish up lecture 2 with the rest of this.